Hi everyone, this is the fourth mini lecture in chapter 13. Today we're going to be talking about T-cell response. So with T-cell response, this is also referred to as cell-mediated immunity. The T-cell reactions are the most complex and diverse in the immune system, and this is because they involve several subsets of T-cells. We're going to go through each of these in this mini lecture. So all of the actions of T cells are dependent on those APCs, those antigen presenting cells, to activate them. And they're restricted because they require some type of MHC recognition. Remember, to recognize that APC, it must actually recognize the self complex here before they can be activated. And all T cells will produce cytokines that have a large spectrum, a wide spectrum of biological effects. And the end result of T cell stimulation is going to be mobilization of other T cells, B cells, and phagocytes. This is table 13.4 in your book, and these show the characteristics of subsets of T cell types. Once again, T cells are sensitized with the antigen MHC complex, and they'll divide and differentiate into one of the subsets below. We have the top four here that are all CD4 receptor cells. These include T helper cells 1, 2, and 17, and then the T regulatory cell. The T helper cell 1 is going to be the one that activates the cell-mediated immunity pathway. It will secrete tumor necrosis factor and interferon gamma. And then we have our T helper cell 2. This is going to be the one that drives B cell proliferation and secretes interleukin 4, 5, and 13. We have our T helper cell 17 that will promote inflammation and secretes interleukin 17. And then the last of our CD4 receptor T cells is our T regulatory cell. And this one will control specific immune response and prevents autoimmunity. The last of our T cell types that can be divided into is the cytotoxic T cells. These are a little different in that they have a CD8 receptor, and they will destroy target cells by lysis. And they are an important part of the destruction of microbes, cancer cells, virus-infected cells. They also play a role in graft rejection. And they require MHC1 for function instead of MHC2, which we'll look at in a little bit. So the T helper cells, once again, there's three types of these T helper cells that all bear that CD4 receptor, and they're critical in regulating the immune reactions. They also involve activating macrophages either directly by receptor contact or indirectly by the release of cytokines. One of these subsets will secrete interleukin 2, which will stimulate the primary growth of T cells, including those cytotoxic T cells. And the one that does interleukin 2, this is that CD4, the CD4 cell. We have ones that will secrete interleukin 4, 5, and 6, and these will stimulate the B cell, and this is your T helper cell 2. And the T helper cells make up about 65% of the T cell population, so they're our most prevalent T cells. We also have the cytotoxic T cells, and these are cells that will kill other cells. They're also referred to as CD8 killer T cells. They target cells that are virally infected, perhaps are cancer cells or cells from other animals and humans. And they have that CD8 receptor, and they bind to that MHC1 complex. Oh, sorry, we went back one. We'll go ahead and go forward to additional cells within this immunity. We have natural killer cells. These ones lack specificity for antigens, so they differ from the other T cells. They circulate through the spleen, blood, and lungs. And they're usually the first killer cells to attack cancer cells and virus-infected cells. Although they're not part of the specific immunity, they are sensitive to interleukin 12 and interferon, so they are stimulated by the specific immune system. We also have other cells with orders to kill. We have something that are called natural killer T cells, and these are actually a hybrid, part killer cell and part T cell. They have T cell receptors for antigens and the ability to release large amounts of cytokines very quickly, which would lead to cell death. The last slide of this lecture is on T cell activation. This should summarize everything that we've covered in this mini lecture. This is table 13.5. At the top, we are looking at the helper cells and the regulatory cells. So what we have here is that APC, which has an antigen on its MHC2 complex. It has bound to a CD4 cell through that antigen and also through that CD4 receptor to the MHC1. We have release of interleukin 2, interferon gamma, and interleukin 4. And what happens is through this stimulation then of the CD4 cell, we get proliferation of T cells. We get those memory CD4 T cells for in case this antigen invades again. We get T helper cell 2 that will produce interleukin 4 and will activate the B cell system here. We get proliferation of the T helper cell 1, which is going to produce tumor necrosis factor and interferon gamma. These then will go on to stimulate macrophages. We also get proliferation of T helper cell 17. These play the role in inflammation, and they secrete interleukin 17. And then we also get this T cell regulatory cell, which will dampen the immune response as appropriate. It's the one that will rein it back in when we have the infection under control. So this is a nice review of all of the different T cell, CD4 cell proliferation. The bottom of this table then goes into the CD8 activation. So we have the CD8 cell here, which will bind to an MHC1 antigen presenting molecule. We will have interleukin 2 that will then also proliferate helper cells. And the CD8 cell, or cytotoxic cell, will then proliferate. It will go on to do memory cells for if this antigen invades again, and it will activate CD8 cells. These CD8 cells then will recognize the infected cell as self, but recognize also that it needs to be destroyed because it's either an infected host cell or perhaps a cancer cell. So it will get to that target cell and it will then secrete perforins and granzymes. Perforins will actually create holes in that membrane of that infected cell. And when it creates these holes, we will have leakage, which will allow the granzymes in. These granzymes will break down proteins of the host cell, and eventually this will then destroy the host cell. So hopefully this table will give you a really good view of what happens either with an APC presenting to a CD4 cell or to a CD8 cell. And this will help out for those of you who are doing the T cell activation on our wiki assignment of this week. The last lecture of this chapter will cover B cell activation.